Hello student, today we will discuss about the genome organization of eukaryotic cell. Here we take the eukaryotic cell of human cell, okay? So at first we have to know what is genome. Genome means all sets of genes of a living cell. So all the sets of the genes which are present in a cell is known as genome. Then what is genes? What is genes? Genes means the nucleotide sequence. The nucleotide sequence which is capable to express is that which can transcribe and form mRNA, which is capable to express. And today's topic is genome organization of eukaryotic cell. Here we take the human cell. Okay, so the eukaryotic cell, where it is the human cell, and there the presence of genome is the genome size of human. In human, if we take haploid cell, then it would be like that, 3 into 10 to the power 9 base pair. When we take the diploid cell, then it would be multiplied by 2. 2 into 3 into 10 to the power 9. This is for diploid cell. And then we will calculate the total length of the genome. Then the genome length. What is the genome length? This is the genome size, and then it is multiplied by distance between two base pair. And what is the genome size? This one. And distance between two base pair, we know that this is 3.4 angstrom. Then we calculate like that. This is 3.4 angstrom. Okay. Then we will convert this angstrom into meter. Then what will happen? Like that. If we convert an angstrom into meter, then we have to multiply it with 10 to the power minus 10. Then it would be like that. Okay. This is I'm just removing this portion. Please student complete all the notes, and then it will be what, like that, 2 into 3 into 10 to the power 9 base pair, and then 3.4, and then it will be 10 to the power minus 10 meter. And if we calculate this portion, it will take 2.04 meter. As we know, the total length of the genome and human cell in a cell, this is 2.04 meter. But in our nucleus, it will be packed. And the nucleus size is? The nucleus size is 10 micrometer. And when the genome size is greater than this, the nucleus size, but it have to be packed within this nucleus. So what we have to do, the cell organize this total genome in a packing system, and this is the genome organization, and there are present different physical levels. We will study about that and how they are packed within this nucleus size. We will thoroughly discuss about all the things. Okay, so let's start the physical level of genome organization. Let's start. Okay, then I'm removing this all the portion. So the levels of how the genome organized within the nucleus. The first level is the nucleosomal model. And here the nucleosomal model, if we want to study the nucleosomal model, then first we have to know what is chromatin. The chromatin is the interaction between DNA and protein. Chromatin means the interaction between DNA and protein. And here the protein is two types. One is histone protein. Another one is non-histone protein. Histone protein is a octameric subunit. It is a octameric subunit. What is octameric? When the subunit is consist of 8 subunit, this is known as octameric subunit. And it is also known as, it is a basic protein. Why it is basic protein? Because this protein has rich of lysine and amino acid. Because those amino acids are basic in nature, thus because of this is a basic protein. And the non-histone protein is acidic or it is neutral type of protein. And the example of the non-histone protein, this is one like condensin or HMG. This is the high mobility group protein. Or another one is topoisomerase. So this is chromatin. So when we discuss about the nucleosomal model, then we discuss about the chromatin, then we have to draw this protein. This is the histone protein. So let's draw the histone protein and then removing that portion, and this is histone protein. As I said that the histone protein is 8 subunit protein, so this one is like that. 1, 2, 3, 4 portion, and another 4 portion 1, 2, 3, 4. This is the octameric histone protein. This is H2A, H2A, H2B, H2B. This is H3, H3, H4. This is H4. This is the histone protein. And there is also present their N terminal. What is? This is N terminal. And when the histone protein is interact with the DNA like that, then this is would be the nucleosomal model. And there the interaction of DNA, the DNA there interact 147 BP DNA is interact with the histone protein, and then this would be nucleosomal model. And we should know that this 147 BP DNA, this is turned the histone protein 1 and 3 and 4 time. This is the turn, not 2 complete turn. This is this type of turn, and we can also say that 1.66 time it turns, is that the DNA turns the histone protein. And here the histone protein, which is H2A, H2B, H3, H4, all are very conserved histone protein, where the histone 3 is most conserved. This histone 3 is most conserved, and all are conserved, but histone 3 is most conserved. And we have to know about the histone protein molecular weight, and it is, the histone protein is molecular weight like 10 kilodalton to 20 kilodalton. And this nucleosomal model is 11 nanometer fiber model. 11 nanometer fiber model. And where we can see that? We can see that at G1 phase, S phase and G2 phase. These all the characteristics of nucleosomal model, and that's all. And the next level of physical level of organization of genome, this is known as the chromatosomal model. So next one is chromatosomal model. So let's discuss about the next one, and I'm removing that. Okay. This is the nucleosomal, and this is the second one is chromatosomal. And it is also seen in G1S and G2 phase, and it is also a 11 nanometer fiber model. Okay. So the chromatosomal model, as you see in that nucleosomal model, where we can see that this is the histone protein, and there is present 147 BP of DNA. But when that 147 BP DNA is added with the linker protein by linker DNA, which connect the another one or the next on nucleosome model, this connection, this addition version is known as H1 protein or linker DNA, because it link another nucleosome. It is also known as H5 protein. All other H2A, H2B, H3, H4 which are present in there, they are conserved, but H1 protein is variable protein. What is? This is a variable protein. So the chromatosomal model interaction, we can see that there are characteristics that there the DNA is present 147 BP DNA, and with that the linker DNA, which is 53 BP DNA. So this is 53 BP and equals to their present 200 BP of DNA. So this is chromatosomal DNA. This is the chromatosomal model of the DNA. Okay, done. And this all the characteristics, where is present H1 protein, which is a variable protein, and there is present 200 BP of DNA. We can see that not like that. We always use like that portion of DNA. So there is also 11 nanometer fiber model. It is also seen in G1S and the G2 phase. And the next, the compaction which is present, it is known as the solenoid model. So next physical level of genome organization is the solenoid model. So the next one is solenoid model. At that solenoid model, we can see that six chromosome when helically arranged, then it would be known as solenoid model. What is that? We just earlier discussed about the nucleosomal model, where we can say that there is present one histone protein and there is linker DNA and 147 of BP DNA. So when we see that 6 nucleosomal model, nucleosome is helically arranged, then it forms solenoid model. How? Like that one. This is 1, this is 2, this is 3, this is 4, 5, 6. And the DNA interact with like that. Like that. So 6 nucleosomal model when interact helically, then it would be solenoid model. So there is present 6 nucleosomal subunit, 1, 2, 3, 4, 5, 6. So this one, and we know that one nucleosomal subunit is 200 pp of DNA. So there is present 6 subunit of nucleosomal DNA. So this is 200 bp of DNA and 6 subunits. So it would be 1200 bp of DNA is present in the solenoid model. So this is all about the solenoid model, and it is 30 nanometer fiber model. This is the compaction. 30 nanometer fiber model. And when you see that, we can see that it is also G1S and the G2 phase. And this is all about the solenoid model. And then we will discuss about the next level of physical level of organization. This is the genome organization. The next one is the scaffold model. And what is scaffold model? So let's discuss about the scaffold model. This is the fourth one. This is the scaffold model. Here we have to know what is scaffold. Scaffold is a non-histone protein. Non-histone protein, as we discussed earlier, it is a non-histone protein. So there will be three examples, and I said it earlier. Non-histone protein. It is a non-histone protein. So we can say that the example of this is condensin. And when the earlier one is that the solenoid model is interact with the scaffold protein, which is the condensin, then it would be the scaffold protein. This is the condensin protein. And when the nucleosomal model and interact with the, and form the solenoid model, and then the solenoid model, the helical conformation is attached with the condensin protein. This is the condensin protein. It is the helical conformation like that, and it is which portion? It is the solenoid portion, okay? Solenoid model. When it is interact with the condensin protein, which is a scaffold protein, then it would be known as the scaffold protein. And here they present this one, the helical conformation like that way. This is not. There is also present in another one. This is the 6th subunit compaction. This one present is this one, and this is helically arranged with the condensin protein. And when it is attached to the condensin protein, this is known as the SAR. What is SAR? SAR means scaffold associated region, and this is known as the scaffold model. This is also 30 nanometer fiber model, and this is also G1S and G2 phase. It is also shown. And then the next level of physical organization, the genome, it is known as chromatid model. So let's discuss about chromatid model. The 5th one is chromatid model. This one, the chromatid model. This chromatid model is shown at mitotic phase. Where? It is shown in the mitotic phase, okay? And at mitotic phase, and they are present on more compaction, okay? So at chromatid model, when the scaffold protein is helically arranged like that, when the scaffold protein condensin is helically arranged like that, condensin protein is helically arranged. And as we know from the scaffold model that the condensin protein when helically arranged, there is also present this like solenoid model. So it is also be like that way, helically arranged, and this helically arranged conformation of DNA and protein interaction is known as chromatid model. This is the helically arranged condensin. It is the scaffold protein, and this is the solenoid protein. Is that the solenoid model? And all this known as chromatid model. When the DNA and protein interaction conformation is like that, then this model is known as the chromatid model. And the next level of physical organization is known as chromatosome model. Is that when the two chromatid is togetherly present and they interact with each other, then this would be chromatosome model. What is like would be that chromatosome model. When the two chromatids is interact with each other or present together, then it would be chromatosome model. Is that when the two chromatid, this is the chromatid model, this is the chromatid model. Then when they are togetherly present, they form this one, and this is chromosome, and this chromosome formation is known as chromatosome model, and this is the more compacted model where we see that this type of chromosome and both strand is known as the sister chromatid, and we always have to think about that chromosome structure is always like that, is that the upper strand always be small, this is called P arm, and the lower arm always long, which is Q arm. This is Q arm, this is P arm, and it should be small and it should be long. And that's all about the physical level of genome organization. Thank you to keep listening my words. Thank you.